Hello viewers, welcome to the course of Introductory Plant Pathology. We will be discussing today the bacteria as the plant pathogens. As we know that bacteria are unicellular prokaryotic microorganisms. They are very simple in nature. As in the eukaryotic organism, they lack the membrane bound nucleus in them, and they are ubiquitous in nature, meaning by they are found everywhere in the earth, and they are very adaptive to the nature, and because of this nature they are ubiquitous in nature. They are omnipresent, we can say, and they are highly adaptable. They can survive the very high temperature, very low temperature. They can survive in the long range of the pH. They can tolerate the oxygen tensions, osmotic and atmospheric pressures. As plant pathogens, they cause very serious disease in our crop plants, and I think there is no plant on this earth which is not being affected by the bacteria. So, we come to the physical factors which influence most to these unicellular simple microorganisms. First, we take up the temperature. Depending on the preferred temperature range, bacteria are divided into three groups. They are psychrophiles, mesophiles and thermophiles. Psychrophiles are cold loving microorganisms having an optimum growth temperature between 0 degree centigrade and 15 degree centigrade. Mesophiles are the moderate temperature loving bacteria. Mesophiles are very important because the majority of the plant pathogenic bacteria comes under this group, because they survive under the optimum temperature range between 25 to 40 degree centigrade. And the thermophiles, they are heat loving microbes, and they are capable of growing at higher temperatures with an optimum above 60 degree centigrade. Some organisms grow even above 100 degree centigrade under pressure also. Then we come to the another physical factor, that is pH. Most bacteria grow best in narrow pH range between 6.5 and 7.5. There are two types of pH loving bacteria. They are acidophiles and alkalinophiles. Acidophiles mean they are acid loving, and they grow at the pH values below the 7, that is mostly at the pH 4. And the alkalinophiles, they are base loving bacteria, and they prefer values of 9 to 10 pH. Then osmotic pressure. Microbes contain approximately 80 to 90% of water, and in placed in a higher solute concentration, they lose water, which causes shrinkage of the cell. We generally call it as a plasmolysis. Some bacteria have adapted so well to high salt concentrations, and these bacteria are called halophiles or salt loving bacteria. Then the oxygen is the physical factor. Microbes that use oxygen for energy yielding purposes are called aerobes. They live in the oxygen bound atmosphere. If they require oxygen for their metabolism, they are called obligate aerobes. So, they cannot live, obligate aerobes cannot live without the oxygen supplied. Often aerobic bacteria have retained the ability to grow without oxygen. These are called facultative anaerobes. And those bacteria that are unable to use oxygen, or rather harmed by it, are known as obligate anaerobes. Then we come to the diseases caused by these pathogenic bacteria in the plants. We see that certain types of symptoms are produced at the time when bacteria infects the plants. So, these are the general types of symptoms I am discussing here. These are the spots on the leaves and fruit, blights or necrosis of the tissues on leaves, stems or tree trunks, rots of any part of the plant, usually the roots or tubers, and wilts due to plugging of vascular tissues. Wilts are very important plant pathogenic bacteria causing diseases, and they actually what they do the plug the vascular tissues which conducts the mineral water and the mineral transportation system of the plant when they are affected by the bacteria, and due to the plugging of vascular tissues the supply of the water translocation is checked completely, and the leaves are showing the drooping type of symptoms and they wilt and die eventually. So, we are discussing here some economically important phytopathogenic bacteria. Actually, in the year 2012, a journal Molecular Plant Pathology has conducted a benchmark survey of the bacterial plant pathogens throughout the world, and after coming out of their results, they noted down certain 10 most important bacteria all over the world which are causing great disease and severity of disease in the crop plants. So, I am here showing you that which are the 10 bacterial diseases causing pathogens which are very important all over the world. They are Pseudomonas syringae, Ralstonia solanacearum, Agrobacterium tumefaciens, Xanthomonas oryzae, Xanthomonas campestris, Xanthomonas axonopodis, Irvinia amylovora, Xylella fastidiosa, Dickia dedentii and Dickia solanae, and lastly the Pectobacterium keratovorum and Pectobacterium atrosepticum. Now, we come to the most important symptoms as we see in the diseased plants. So, first of all, we are seeing here the bacterial wilt of eggplant. Eggplant is our brinjal. So, you are seeing on the both the sides of they are having resistant lines of the eggplants, and in the middle we are having the susceptible plants. S is denoted by susceptibility and R is resistance. So, as we look into the resistant plants are growing very well, whereas the susceptible plants are all died due to the bacterial wilt infection. In the second photograph, as you are seeing here, the bacterial canker is showing the gummosis or the gumming in Prunus species. Here, after the bacterial infection, the spots emerges out from the trunk of the trees, and the margins of the spots produce gum, which you are seeing here. Then when such type of infected plants are exposed and the cambiums are showing here, you see they are showing the cambiums are showing the necrotic and discolored areas. Again, when we look into the bacteria under the electron microscope, how they look like we are presenting here to you, that these are the most important bacteria. As I told you, this is the Agrobacterium, this is the Irvinia, this is Pseudomonas, and this is the Xanthomonas. They are very dreaded disease causing bacteria. Then we come to the history of the bacteria. As we all know that history show us that how the science of bacteriology developed in the past, what type of problems were encountered by our scientists in the past, and how they tackled those problems by their ingenuity and their intellect. So, this informs about the contributions made in that field as a source for the growth of the knowledge. It enables us to appreciate what bacterial science is about and how it is conducted. And in the manner they tackled the disease, we study the history of the bacterial plant pathogens. This was a Dutch scientist who first of all, Anton von Leeuwenhoek, in the 1675, he first of all discovered, or rather he developed a compound microscope of his own, and he first of all saw the bacteria by his eyes through his developed microscope. He first visualized bacteria in 1683 under his microscope. Only a little over 100 years ago, a bacterium was first implicated as a causal agent in the plant disease. So, it is not very early that we have come to know that bacterial pathogens are causing very, very important and serious diseases. It is only 100 years before we could get this idea. This is the picture showing the Anton von Leeuwenhoek, and you see this is the microscope developed by him, and he saw first of all the bacteria of this type, it is showing in the photograph, and he discovered the bacteria first in the dental tartar, including a motile bacillus, a micrococcus and a motile spirochete. Then American botanist T.J. Burrill in 1873 proved that bacterial nature of fire blight disease of apple and pear. And in 1878, bacterium Erwinia amylovora was found associated with this fire blight disease. By the year 1900, Dr. E. F. Smith established the role of bacteria as plant pathogens. Dr. Smith is now considered as the father of bacterial phytopathology. He worked on bacterial wilt of cucurbits and crown gall diseases. And this is the photographs of the crusaders of bacteriology. Here it is the photograph of Thomas Jonathan Burrill, who worked on the fire blight disease of pear and apple, and he showed us that the bacterial fire blight is caused by bacterial pathogens. And then the Robert Koch, who gave us the Koch postulates to propound it, that by isolating the pathogen and culturing it on the artificial media, and again re-inoculating the pathogen on the plant, and again isolating the same identical pathogen is the process by which we can establish that this disease is caused by this particular type of plant pathogen or particular type of plant bacteria. I am showing here the first bacterial disease, the fire blight caused by Erwinia amylovora, and in 1885 J.C. Arthur was able to isolate a bacterium from diseased plants, cultured it, and then inoculated the same host to reproduce a naturally occurring disease. He recovered it subsequently from diseased tissue, fulfilling what is known as Koch's postulates. And here you can see the devastation of the fire blight caused by Erwinia amylovora in the apple orchard. Bacteria are among the microbes that successively colonize plants. Exactly, they grow very slowly on the plants, and slowly they colonize whole of the plant parts. A large population of bacteria are visible as aggregates on the liquid as biofilms and viscous suspensions on the plugging of the plant vessels when they cause wilt. Plant pathogenic bacteria cause many serious diseases, but we can say that bacterial diseases are fewer in comparison to the fungal and viral diseases. Then we come to the morphology and cell structure of bacteria. Bacteria generally range from 1 to 2 μm in size. Cell wall is made up of mucopeptide in place of cellulose, as in case of eukaryotic organisms. They lack organelles like mitochondria, chloroplast, Golgi body and lysosomes. Ribosomes are of 70S and not of 80S types as in the eukaryotes. Mesosomes are the sites of respiration and membrane lipid synthesis. Photosynthesis in bacteria is through vesicles and lamellae. Then morphology, it is the study of the size, shape and structure of the bacteria and how the bacterial cells are arranged, and this type of morphology actually help the scientists to classify them in a particular type of order by which we can easily study the organisms and we can classify in a different classes and phylum. And in this type of things we are discussing here the shapes of bacteria first. Bacterial cells are spherical, ellipsoidal or oval, and they are called as cocci. Cylindrical or rod shaped are bacilli, spiral or helical are spirali, and some species have varieties of shapes and thus termed as pleomorphic in nature. Bacteria associated with plants can be seen with conventional microscope at 400x to 1000x magnification. Bacteria without detectable appendages can be effective pathogens, and on nutritive media plant pathogens usually grow more slowly than non-pathogenic bacteria. This is the most important sign of pathogenic bacteria, and with optimal temperatures of 20 to 30 degrees centigrade they grow very well. Now we come to the arrangement of bacterial cells. Bacterial cells are arranged in a characteristic manner of the particular species. All species are having a characteristic type of cell arrangement, and the typical pattern of cell arrangement in different bacteria is an important characteristic used in the bacterial identification. This is the cell grouping or arrangement in bacilli. In bacilli there are monobacilli, they are single rod shaped bacterial cell, for example monobacillus. Then diplobacilli, bacilli are arranged in a pair of two cells, for example Bacillus subtilis. Streptobacilli, cells are arranged in a chain, for example Lactobacillus bucarius. And then palisade, group of cells lined side by side like matchsticks in a matchbox called a palisade arrangement. Here we can see the different types of shapes of bacterial cells. Here these are the coccus, single cell. Then in the pairs they are known as diplococci. In the chains they are called streptococci. In cluster staphylococci, and like that. There are so many types of shapes, and we can see here the different types of vibrios. They are comma shaped in nature. Then the curved spirillum spirochetes are curved. Then spirilla, they are bottle opener type of shape is there. Spirochetes are the filamentous in nature, and these type of different shapes and morphological types are found in bacterial cells. Some rod shaped bacteria called vibrio are slightly curved or comma shaped. Others can be spiral shaped called spirilla, or tightly coiled called spirochetes. A small number of species even have tetrahedral or cuboidal shapes. This is the typical, see the bacterial cell structure. As I told you earlier that it does not have the perfect nucleus as is found in the eukaryotic organisms. This is the cell wall which is of the murine. This is the capsule, outer capsule. These are the plasmids, cytoplasmic membrane. Then 70S ribosomes are there which work for their protein manufacturing, and then depot substances, and they are volutin and glycogen, and certain granules are there, and there is also a pili in all bacteria which help them to attach to a particular surface. outer membrane is only of in the gram negative bacteria we find the outer membrane, otherwise in gram positive bacteria it is absent, and then flagella are present which help them to move or help them in the motility of such type of bacteria. Now we come to that how the bacteria look like under scanning electron micrograph. You see the cocci type of bacterial colony are growing in a particular medium here. Then as we have discussed about the devastations and severe disease causing plant pathogens, but at the same time they are also useful organisms, and they give us so many types of products we use daily in our lives. So here we are discussing about the useful bacterial pathogens. See for instance Agrobacterium radiobacter. They are useful for our biological control. They control so many plant pathogenic diseases. They help in the biodegradation of atrazine. Then Agrobacterium tumefaciens, they function in the plasmid vector for plant transformation. This is very important pathogen, as we see in the crown galls. Crown galls are just like our cancerous cells in the human beings, and the cancer research is first going on in the crown gall bacteria, and by the mechanism of the crown gall our scientists are trying to manage the cancer disease in the human beings also. Since they are very important in such type of research material, so they help in the transformation of the plants. Erwinia amylovora is a source of harpin, an elicitor of disease resistance in plants. The Xanthomonas campestris, former species campestris, is a xanthan gum. It produces a polysaccharide used in food production, in agriculture, cosmetics and pharmaceuticals, and several other plant associated bacteria are there which help us in producing restricting endonucleases, enzymes used for specific cutting of DNA in scientific research. Viewers, today we discussed about the morphology and cell structure of bacteria and about the bacterial history. As we know that bacteria are unicellular simple microorganisms, and they are very adapted to the nature, as we found them in all types of temperatures, pH ranges, under oxygen stresses, osmotic pressures, and we have studied that what are the ranges in which they can grow. Secondly, we have discussed about the different morphological structures in the bacterial cells, and we have seen that a number of morphological structures are there in plant pathogenic bacteria. And beside this, we have also discussed about the most important plant pathogens as bacteria, and we have discussed about a survey conducted in the 2012 and found out that what are the 10 most important bacterial plant pathogens in the world today. We have also discussed about the useful bacteria which are producing a large number of products and which are helping us in our research towards medicinal research, towards the cancer fighting research, and so on and so forth. Thank you.